Hello, biology students. We're going to talk about the second step of making proteins, protein synthesis, translation. And I remember it's second because it has that later in translation. So we talked last time about how DNA becomes a copy of itself through transcription, mRNA. Those are both nucleic acids. Now we're going to talk about, we're talking about protein synthesis, how we make a protein. The last step is how we get to the protein, which is a whole new type of macromolecule. These are nucleic acids. This is a different thing, proteins. So because we're changing from the language of nucleic acids to proteins, we call it translation. I'm changing languages. I'm going from nucleotides to amino acids. So that's really, really important here. And the way we do that is by reading a code. The code is called a codon. A codon is 3 letters in the mRNA that we use to code or translate into a building block of proteins called amino acids. So 3 letters like CGA or GCA, all right? There's lots of combinations of 3 letters. Each of those 3-letter combinations will make an amino acid. And we have charts in class called codon charts that tell us which amino acid goes to which 3-letter combination. So this is really important. I need that mRNA in the middle because it allows me to then figure out which amino acid I need to make my proteins. So how does this process work? Well, we left off last time when DNA made a copy of mRNA, that's this flat type of RNA, but unlike DNA, mRNA can leave the nucleus. All right, so transcription is in the nucleus. Now mRNA is leaving the nucleus and entering that gel-like substance called the cytoplasm. And in the cytoplasm, what we learned about when we did cells a long time ago, those little dots that looked like sesames or freckles in the cytoplasm, ribosomes, we're looking at them way more close up. Now they look like 2 pieces of hamburger buns, right? And they're going to clamp down on top of the ribbon. All right, so the ribosome is going to just chomp onto the long string of this mRNA. And what it will do, ribosome reads the mRNA. It's going to read along this message. Ribosome reads the message. So where does it start to read? Well, at a codon called the start codon. And so they will find a 3-letter code. Remember, codon's 3 letters. And it will clamp down right there. All right. Notice that there's this weird thing coming in also that looks like a clover shape. We're going to talk about that right now. What's that weird clover thing? Well, that weird clover thing is called a tRNA because that clover shape also looks like the letter T. But it is not DNA. It's not a double helix. It's a RNA just folded up on itself. And on the top of this RNA is an amino acid. And because it's this funny T shape, and it also has something kind of on the back of it, I call it a truck. A truck that trucks in amino acids, the building blocks of proteins. So these things are really important. Why? Well, because they're going to bind to the ribosome. And they're going to bring the right amino acid to the right location on the message. So here we have a ribosome, and it's going to act like a garage or a docking station. All right, so this is the garage, and this truck, right, will bind to the right part of the message. Notice that there's 3 letters here on the red, 3 letters here on the red, and that matches the 3 letters and 3 letters here using base pairing rules. And at the top of each of these tRNAs is an amino acid. These are the same amino acids. If I looked up these red letters in a codon chart, this is the amino acid methionine or MET that I would see on a codon chart. If I looked up GUC on a codon chart, I would see valine or VAL on a codon chart as the correct amino acid. So these tRNAs are the trucks that bring in the right amino acid to the right part of this mRNA message. It's reading it. The part that it uses to attach to the right place is called the anticodon. The anticodon is on the tRNA. Here it is. Next, as I bring in more amino acids to this garage or the ribosome, I'm reading the message and they begin to form a long, long chain. Remember, amino acids, we decided, bind together to form a protein. We even decided last set of notes that the bonds between amino acids are called peptide bonds. And so we're going to call a chain of those peptide bonded amino acids a polypeptide. That's another word or a synonym for proteins. Eventually, I'm going to reach a signal that says, stop. We're done reading the message. There's no more message. Don't keep going. And that stop says to the ribosome, release. And the two bun-like things come apart, and the protein or polypeptide is released. That means we're done. And proteins make things like eye color, our bones, enzymes. And so this is maybe potentially an enzyme or a thing for my eye color. We've now learned about a couple types of RNA. What were they? Well, we had mRNA and tRNA. Our mRNA was like the ingredients list or a recipe card. And remember, we saw it in the nucleus and in the cytoplasm. One for transcription and one for translation. We also saw tRNA today for the first time. tRNA was only in the cytoplasm during translation. And we called it like a truck because remember, it goes into the garage, the ribosome, and it brings in the right amino acids to the right spot. Notice they're different shape-wise, and you're going to have to be able to recognize that this one's more like a ribbon and this one is more like a clover. Lastly, what was the big idea of this whole thing? Well, in translation, I took my mRNA message and I translated it into amino acids at the ribosome. I read it by reading codons, which were the language that allowed the correct amino acid to be trucked in by tRNA. That was hard, but great job, guys.